Imagine your body not as a single solid thing, but as a bustling microscopic metropolis.
Right. Trillions of citizens just moving around.
Exactly. Trillions of citizens. And just like in any major city, who you live next to changes absolutely everything about your day.
Oh, totally.
Like if you live next to a bakery, you smell fresh bread. But if you live next to a nightclub, you're not getting any sleep.
Yeah, your environment dictates your behavior.
Right. And the exact same principle is true for our cells. A cell's immediate neighbors dictate its behavior, its survival, and, well, ultimately, whether a tissue stays healthy or falls to disease.
Which is such a crucial concept.
It really is. So welcome to this deep dive. Our mission today is to explore a groundbreaking April 2026 paper from Nature Communications.
By Cellular and colleagues.
Yeah, that's the one. And they have essentially cracked the code on how to mathematically map these cellular neighborhoods. Their work really changes the fundamental way we track disease progression.
It is a truly phenomenal piece of work. I mean, for a long time in spatial biology, figuring out which cells actually prefer to interact with one another has been this massive computational headache.
Because it's not as simple as just looking at a slide, right?
Not at all. So the researchers recognized this gap. They conducted this comprehensive study evaluating how we currently map neighborhood dynamics.
And they didn't just look at one thing. They analyzed simulated computer tissues, triple-negative breast cancer data sets, and even models of myocardial infarction.
Right, heart attacks.
Yeah.
And what they found was that most of our existing tools are, well, they're fundamentally flawed.
Which is a huge claim.
It is. But it led them to develop a highly optimized pipeline to correct these mathematical blind spots.
Okay, so I want to get right into that new pipeline, which they call COZI. But to understand why it's so revolutionary, we first need to look at the baseline.
Right, the fundamentals.
Yeah. The core concept here is neighbor preference, or NEP. It's basically asking, what does a cell of type A actually see in its local environment?
Like, are those infiltrating immune cells actively attracted to specific tumor cells? Or, are they just randomly bumping into them?
Exactly.
Yeah.
That distinction between active biological attraction and just random chance, that's the entire foundation of spatial biology.
It is. And the reason it's so incredibly difficult to calculate comes down to what we call cell abundance bias.
Right. So let's unpack that. This is basically the statistical inevitability of dense tissues, isn't it?
Yeah, pretty much. Think about a tumor core that is, say, 90% malignant cells.
Okay.
Any infiltrating immune cell that manages to get in there is going to be completely surrounded by tumor cells.
Because there's just so many of them.
Exactly. It doesn't necessarily mean there is a strong biological homing mechanism or like a specific cellular attraction. It's just the physical reality of the space.
You're just surrounded by the majority. So this is exactly why visual inspection or even just basic counting is useless here.
Totally useless.
Yeah.
If you just trust the raw counts, you'll assume there's this massive biological preference.
When in reality, it's just random chance based on abundance.
Right. So to mathematically prove that an interaction is biologically meaningful, NEP methods have to utilize a strict three-step algorithmic process.
Okay, let's go through those 3 steps. The first step being neighborhood definition, meaning we have to draw the borders around an index cell.
Correct. And there are traditionally 2 ways to do this.
Right. You can use a fixed Euclidean distance. So that's literally drawing a physical radius of, say, 20 micrometers around the cell.
Yeah, like drawing a circle on a map.
But looking at the density of some of these tissues, Euclidean distance seems, I don't know, incredibly arbitrary.
It can be, yeah.
Because if a tissue area is sparse, a 20 micrometer circle might capture 0 neighbors. So the alternative is graph-based methods like Delaunay triangulation or k-nearest neighbors. Those must be superior because they look at actual topological connections, right?
That is the prevailing consensus, yeah. And it's what the authors focus on. Graph-based methods, particularly Delaunay triangulation, create a web of connections based on immediate proximity.
Regardless of the absolute physical distance.
Exactly. It ensures that every cell is connected to its true adjacent neighbors, forming a reliable spatial network.
Okay, so once that network is drawn, we move to the second step, which is neighbor quantification.
Right, which is just tallying up who is connected to who within that established network.
Sounds simple enough.
You'd think so. But the critical variable here is directionality. You can quantify neighbors non-directionally.
Meaning you just log that cell A and cell B share an edge.
Right. Or you can quantify bidirectionally, which separates the perspectives.
Oh, okay. How does that work?
It asks, From the perspective of cell A, how many cell Bs are neighbors? And then it flips it. From the perspective of cell B, how many cell As are neighbors?
That separation of perspective seems like it would be absolutely vital for understanding things like immune cell attacks.
It's crucial. But even once we have those counts, we still have to figure out if that clustering actually means anything.
Otherwise, we are right back to the cell abundance bias problem we talked about.
Exactly. Which brings us to the third and most computationally heavy step, NEP scoring.
Right. And to rule out abundance bias, they use something called permutation testing to create a null model.
Yeah. We take the tissue network and we keep the exact physical locations of every single cell intact.
Okay, so the map stays the same.
Right. But we randomly shuffle their identities. We turn a T cell into a macrophage. We turn a tumor cell into a fibroblast.
So you're basically scrambling the labels.
We scramble the labels thousands of times in a computer simulation.
So you are basically asking the algorithm to simulate what the tissue would look like if nobody cared who they lived next to.
Precisely. You are creating a perfectly randomized baseline of the neighborhood. That randomized baseline is the null distribution.
Okay, I follow.
We then take our actual observed neighbor count and compare it to that null distribution.
And if our observed count is significantly higher than the random simulations, we have mathematical proof of an active attraction, a positive neighbor preference.
Exactly. And if it's significantly lower, we have proof of active avoidance.
Okay, so let's do a quick recap here. The 3 steps are define the borders, quantify the connections, and score them against a randomized baseline.
That's a perfect summary.
So think about your own neighborhood for a second. If we ran an NEP analysis on you, who would be your statistically significant neighbor? Is there a coffee shop you're actively drawn to, or is there a neighbor you actively avoid?
That's a great way to visualize them.
But here is my question. If spatial biology already has these 3 mathematical steps established, and we have all these software tools calculating these networks, why are the results so inconsistent across the board?
That is the big question.
Because the paper highlights that widely used tools with names like histoCAT, CA, Giotto, and SciMap, they're completely failing to agree when looking at the exact same tissue samples.
And that inconsistency is a massive liability for the field. To figure out why these tools were disagreeing, Schiller's team had to establish an absolute ground truth.
Right, because in real biological tissue, it's too messy. You never truly know the ground truth.
Exactly. So they built what they call in silico tissues, or ISTs.
These are the simulated computer tissues.
Yes, they basically played God.
Right.
They generated tissues with tens of thousands of cells and explicitly programmed the interaction affinities.
So they knew, with 100% mathematical certainty, exactly how much cell type A preferred cell type B.
That's the beauty of the ISTs. You have a perfect answer key to grade the software tools against.
And what happened when they tested the old tools?
When they fed this simulated data into the existing tools, the results revealed 2 major mathematical blind spots that have been holding the field back.
Wow, 2 huge blind spots. Let's hit the first one.
The first is the categorical versus continuous problem. Older tools, like histoCAT and IMCR tools, utilize categorical scoring.
Meaning they look at the permutation testing and simply output a 1, a 0, or a -1.
Right, meaning the interaction is significant, it's neutral, or it's avoiding.
Which immediately presents a massive limitation when you are studying disease progression. Well, if you are tracking a tumor over time, you aren't just looking for a simple yes or no on whether immune cells are interacting with it. You are looking for gradients.
Oh, absolutely.
You might have a healthy tissue with a weak preference and a diseased tissue with a massively strong preference, but a categorical tool just rounds them both to a 1.
It just yells, yes, they interact, for both scenarios.
Right. Completely blinding the researcher to the fact that the interaction is actively intensifying.
It flattens the biological reality.
Yeah.
To see the gradient of a disease, you need a continuous score.
Like a z-score.
Exactly. You need an output like a z-score, which gives you a precise statistical magnitude, like a 3.2 versus a 15.8.
Okay, so categorical scoring is definitely a problem. But the paper said the simulations revealed an even more destructive pitfall.
Yeah, they call it the directionality dilemma.
Right, the concept of asymmetric cell adjacencies. I've always thought of this through the lens of a paparazzi analogy.
Oh, I like this one. Explain it for our listeners.
Okay, so imagine a specific type of immune cell acting as the paparazzi and a rare tumor cell as the celebrity. The paparazzi cell desperately wants to be near the celebrity.
Obviously.
So if you calculate the preference from the paparazzi's perspective, it has a massive 100% preference for the celebrity. But if you flip the perspective...
The celebrity cell couldn't care less.
Exactly. It has zero preference for the paparazzi. It's just trying to mind its own business. The relationship is totally asymmetrical.
And yet, when Schiller's team ran the ground truth simulations, they found that highly respected spatial analysis tools entirely missed this asymmetry.
Wait, really? Tools that thousands of scientists rely on?
Yes. They assigned the paparazzi and the celebrity the exact same neighbor preference score.
But how does the underlying math fail so spectacularly on that? If they are using bidirectional counting, like we discussed earlier, shouldn't they inherently capture the different perspectives?
You would think so, but it all unravels during the normalization step.
Oh, the normalization.
Yeah. Many of these tools rely on what's called total normalization. Let's apply this to your paparazzi analogy.
Okay.
Say you have 100 celebrity cells in a tissue sample, but because the paparazzi cells are rare, only 5 of those celebrities are actually being actively swarmed.
Right. Just a small fraction.
Total normalization takes the total number of paparazzi connections and divides it by all 100 celebrity cells in the entire tissue.
So it includes the 95 celebrity cells that are sitting completely alone in the denominator.
Exactly.
Wow. That heavily dilutes the math. It shrinks the fraction to near zero, and the local paparazzi swarm gets completely washed out in the broad tissue average.
It destroys the signal. To preserve the directionality of the interaction, the authors argue you have to use conditional normalization.
Conditional normalization. Okay, what does that mean practically?
This means you divide the connections only by the index cells that have at least one neighbor of the target type.
So you only count the celebrity cells that are actively engaged with a paparazzi cell.
Exactly. When they analyze tools using total normalization, like SEA and Squidpy, those tools were completely blind to the directionality. They just output blurry, non-specific trends.
So, quick recap again. Categorical scores miss the nuance, and total normalization misses the direction.
Spot on.
But wait, let me challenge the methodology there for a second.
Go for it.
If we use conditional normalization, meaning we only look at the index cells that actually have at least one neighbor, aren't we intentionally blinding ourselves to the broader context?
What do you mean?
Well, by throwing out all the zero interaction cells, don't we lose the macro view of the tissue? If only 5 out of 100 cells are interacting, but we conditionally normalize to only look at those 5, we might trick ourselves into thinking it's a massive tissue-wide event when it's actually highly localized.
That is an excellent critique.
Yeah.
And it is the exact trap that a conditional score alone would create.
Right, because a z-score tells you the statistical strength of a preference among the active cells.
But you are absolutely right. A massive z-score of 50 is biologically misleading if only 2 cells out of 10,000 are participating.
Yeah, that's what I was thinking.
The authors anticipated this, which is why the tool they built doesn't just output a single number.
Which brings us to the real innovation of the paper, COZI, or the conditional z-score.
Yes. They realized the optimal mathematical pipeline didn't exist in one place, so they synthesized it.
So what's fascinating here is that COZI is elegant because it optimizes the flow of data, right?
Exactly. It strictly enforces bidirectional counting so we capture the paparazzi perspective. It strictly uses conditional normalization so we don't dilute the local signal.
And it outputs a continuous z-score so we can measure the gradient of the strength.
And, to answer your critique, it pairs every single z-score with a new metric called the CCR, the conditional cell ratio.
Ah, the CCR. So the CCR is the macro view. It tells you the exact fraction of index cells that are actually participating in the interaction.
Exactly. The CCR grounds the math in physical reality.
Okay, I love this. So COZI gives you the z-score for the localized strength of the event, and the CCR for the physical scale of the event across the whole tissue.
Yes.
I think a good visual mnemonic for you listening is to imagine putting on a pair of COZI glasses.
COZI glasses, I like that.
The lenses use conditional filters to cut through the blinding glare of all the non-interacting cells, showing you the ratio, the CCR, of cells actually interacting.
Right, how much of the frame is lit up.
Exactly. While a glowing z-score tells you exactly how strong that interaction is in those specific spots.
That is a highly accurate way to conceptualize the data output. And the researchers didn't just stop at computer simulation.
Right, they had to prove it in the real world.
To prove that COZI's polarizing filter actually reveals biological truths that other tools miss, they applied it to intensely complex clinical data sets.
Let's look at the first clinical application, triple-negative breast cancer, or TNBC.
This is a notoriously aggressive form of cancer.
And spatial biology is critical here for understanding immune infiltration, right?
Oh, absolutely. We need to know if the patient's immune system is successfully penetrating the tumor islands, or if it's being locked out in the surrounding stroma.
So how did the older tools perform on this data set?
Well, when the researchers ran the TNBC dataset through older algorithms like SEA, which relies on that flawed total normalization, the output was frustratingly vague.
Vague.
The older tools broadly categorized the tumor microenvironments as either mixed or compartmentalized.
Right, because total normalization washed out the subtle, localized clusters of immune cells interacting with the tumor boundaries.
Right, but when they applied COZI, the resolution of the data just snapped into focus.
It could see the nuance.
Exactly.
Yeah.
COZI was able to mathematically differentiate the directionality of the infiltration because it uses both the continuous z-score and the CCR. It could separate the tumors into distinct evolutionary stages of attack.
Wow.
It could identify early infiltration, where you have a small vanguard of immune cells pushing into a tumor cluster.
And it could mathematically distinguish that from advanced infiltration, where the immune cells have completely surrounded and permeated the malignant cells. That distinction is paramount for immunotherapy. I mean, if a drug is supposed to drive immune cells into the tumor core, a tool that just says the tissue is mixed doesn't tell you if the drug is working at all.
No, it doesn't.
But COZI mathematically quantifies the exact state of the battle. It shows you the front line.
It really does.
Which translates perfectly to their second clinical case, which was tracking the healing process after a myocardial infarction or a heart attack in a mouse model.
This is a fascinating time series data set. They mapped the heart tissue at 4 hours, 24 hours, and 48 hours post-infarction.
Because when cardiac tissue dies, the immune system orchestrates this highly choreographed cleanup and repair operation.
Right. And COZI was able to objectively map this timeline.
It proved quantitatively that neutrophils, those are the rapid responders, dominate the injury site at the 4-hour mark.
Yeah. Then it showed the shift at 24 and 48 hours, where CCR2 positive monocytes take over the operation.
But the most impressive part wasn't just identifying the cells, it was mapping their spatial route.
The spatial route is everything in this context.
Yeah. COZI proved these cells were migrating directionally through the endocardium, the inner layer of the heart.
Because if you know where the immune cells are coming from, you can infer the chemokine gradients, the chemical signals that are guiding them.
And the older methods couldn't do that.
The older methods struggled to definitively highlight this route because cardiac tissue is a dense, complex web of varying cell types. COZI's conditional z-score cut right through that density.
But let me push back here. Couldn't a highly trained pathologist just look at a slide at the 4-hour mark and see the neutrophils gathering at the endocardium? Why do we need a complex algorithm for this?
Well, human observation doesn't scale, and it's highly subjective. The human eye is easily tricked by abundance.
Okay.
To truly prove COZI's superiority over human observation, the authors took it a step further, using machine learning.
Okay, this is where we get into the random forest classifier.
Yes. The authors used random forest models to classify the tissue samples based purely on the spatial metrics generated by the different software tools.
And they evaluated the performance using F1 scores.
Right.
And for you listening, just for context, an F1 score is a stringent statistical metric that balances precision and recall. It tells you how accurately a model can identify the true biological state without being distracted by false positives.
A great explanation. And when the machine learning model was fed data from the older, totally normalized tools, its F1 scores were mediocre.
Because the blurry data led to blurry predictions.
Exactly. But when the model was trained on COZI's conditional z-scores and CCR metrics, it achieved near-perfect F1 scores.
Near perfect.
Yeah. It could flawlessly predict the time point of the infarction based solely on the neighborhood preferences of the cells.
So it removes the human bias entirely.
Completely.
It allows researchers to process thousands of tissue samples across global clinical trials and objectively say, based on the conditional z-scores, this specific immunotherapy is accelerating immune infiltration by 20% compared to the baseline.
You simply cannot do that with a tool that spits out categorical ones and zeros.
No, you can't. It elevates spatial biology from an observational science to a truly quantitative predictive science.
It really is a paradigm shift.
So let's synthesize this whole journey. What does this all mean for us? We've journeyed from understanding the basic biological need to map cellular neighborhoods to uncovering how total normalization and categorical scoring were creating massive mathematical blind spots.
Right, the paparazzi problem.
Exactly. And we've seen how COZI fixes those blind spots by focusing only on the active participants using conditional normalization.
Calculating the strength with a continuous z-score.
And tracking the scale with the conditional cell ratio, the CCR. It's not just an algorithmic update. It's a new lens that revealed the directional vanguard of immune cells in breast cancer and the precise spatial highways of cardiac repair.
If we connect this to the bigger picture, spatial biology is rapidly moving toward massive whole organ 3D datasets.
Yeah, we're zooming out.
And as we develop advanced immunotherapies, we are no longer just asking, did the tumor shrink?
Right.
We're asking pharmacodynamic questions.
Like, did this specific drug actively alter the neighbor preference of a patient's T cells?
Exactly. Did it force them to seek out and attach to the malignant cells? Tools like COZI provide the exact mathematical framework needed to verify that those drugs are successfully rewriting the spatial rules of the tissue.
It's the ultimate form of precision medicine, and it leaves us with a truly fascinating, kind of mind-bending concept to think about.
Oh, I'm ready.
If algorithms like COZI can precisely map the spatial preferences and directional attractions of every cell type in the human body, could we eventually use this data for cellular urban planning?
Cellular urban planning. Wow.
Yeah, right. If we mathematically decode exactly whose stem cells want to live next to, could we engineer synthetic organs from scratch simply by manipulating their neighbor preferences and letting them build themselves?
That is incredible to think about.
It fundamentally changes how you view biology. So, I challenge you listening, the next time you are standing in a crowded public space, take a look at the people around you. Who did you choose to stand next to? Who did you avoid? And what are the invisible statistical neighbor preferences silently guiding our own human architecture?
A brilliant question to leave off on. The patterns at the microscopic level often echo the macroscopic world in surprising ways.
They really do. Well, until next time, keep diving deep.